So let me quickly start and introduce you to our today's guest, Lilian Weber. Or maybe first of all, for those of you who have not participated during the last weeks, my name is Lilian. Together with David from Lübeck University, we are organizing this lecture series on control this summer together with Timo Thorstenschmidt and Felix Brandenburg. So today we have Lilian Weber as our guest from Oxford University. Some background information. Lilian studied psychology at Marburg University together with physics at that time. And after her diploma in Marburg, she went on to do her PhD at ETH Zurich in the translational and neuromodeling group by causal and Stefan. After her PhD, Lily continued at Oxford University to do the first postdoc in the group of Lawrence Hunt on cognitive computation and neuroscience. And I think about like 2 years ago, she switched to do a second postdoc position, took over a second postdoc position in the group by Miriam and Flügge, also at Oxford University, which is on motivation, decision, and neurostimulation. And we already heard before that she will present some data and findings from this time. Luckily, Lilian Weber received a professorship just a couple of months ago and took over or will take over this position in August. So she will be very close soon. Will be at the University of Osnabrück, from Oxford to Osnabrück. So it will be in close proximity. And maybe we will already hear some of the plans and ideas that you will continue on in a new position in Osnabrück. And so we are very much looking forward to your talk and the discussion, and I head over to you. Thanks for joining.
Thank you, Zoran. That was a very kind introduction. I cannot see anyone. So if you want to interrupt me, ask questions, or let me know that I'm running out of time, please shout. Okay. Thanks everyone for coming to my talk, which is on the neuromechanisms of control and emotional decision making. And so I have been thinking about how my work relates to control. And I think, in emotional context, we face several challenges when we want to make good decisions. And one of these challenges is quite basic, is the decision of whether we want to interact with an emotionally charged stimulus at all. So we've all seen this, a grumpy toddler, and some of us even see this multiple times in a day. And so I personally always prefer to not interact with children when they're like this. And of course, more generally, we have this tendency to prefer to avoid negatively charged stimuli and approach the positive stuff. And that's what we call automatic approach avoid biases. Now, most of the time when I ignore my children when they're like this, that's actually a good strategy. But sometimes, sadly, it doesn't work that way, and everything just becomes worse. And then, I need to bite the bullet and interact with them to finally settle them down. By the way, these are not my children. Mine would look much more dramatic in the situation. So anyway, I think, what is the right action in any situation and in context can change over time, and we can learn, what the right way to solve these situations is, whether to approach or avoid, by, looking at the feedback we get. So apart from this challenge, these automatic biases we have, and the fact that the right action might change over time, there's a more fundamental challenge in emotional situation, which is that emotions generally change over time. And in fact, emotional contexts are quite volatile. And on top of that, emotional expressions, emotional stimuli can be quite ambiguous or even noisy. So when you want to judge the mood of someone based on their facial expressions, for example, you want to see how I feel at the moment, then it's usually a good idea to look at them, for a little while, and integrate over multiple facial expressions and to get at a more precise estimate of their mood. But also depending on the person, you might want to look out for really sudden shifts in their mood. So those are two challenges. There's obviously many, many more, but those are the two that I happen to have worked on quite recently. And I will frame them as, situations where we need to exert control, partly because that will qualify me to speak in this seminar series. So I will present today two studies that look at these two challenges. The first one is an ultrasound study. So there we targeted the amygdala with a transcranial focused ultrasound to determine its causal role in driving these biases. And the second study I'll talk about is a study with a pharmacological intervention. So there we used the drug ketamine to change how people integrate evidence about emotional expressions over time. And the general approach that I take in these studies and also more generally in my work is that I like to use causal interventions on the neural level to determine, how specific receptors or specific brain regions are causally involved in certain cognitive processes, and then I measure, behavior and fMRI and EEG. And then I also use cognitive models to understand the effect that these perturbations have on the level of cognitive computations. I will not talk about this much today, but I have recently thought a little bit about both of these domains. So how do we integrate evidence in dynamic environments? That's the preprint. And then also, how do we model affective stuff or reward in humans? And that's the TICS paper that just came out last week. The other thing that's worth noting before I start is that these two projects are the two main experimental studies that I've run during my postdocs. The amygdala study is worked together with Miriam Klein-Flügge here in Oxford and her lab, and the ketamine study, even though it's still ongoing, was started during my first postdoc with Florence Hunt several years ago. And that already tells you that these studies have been a little bit painful, practically speaking. And even more so, I want to acknowledge this absolute hero here, Maruna Rascu, who's a PhD student with Miriam and me, because she basically soldiered through both of these big studies with me. So shout out to her. Okay, let's first talk about the amygdala study and maybe talk about why we were interested in understanding the causal role of the amygdala in the first place. We know obviously that is a key region for emotion processing, has been involved in lots of processes relevant in emotional contexts. And we also know that it's changed in mental health conditions, and that is probably causally involved in some of these. We are particularly looking at the basolateral amygdala, and that is extremely interesting because it's kind of an interface between prefrontal cortex, but then also brainstem and hypothalamus, etc., etc. So it kind of has connections, structured connections to many different networks in the brain. When you look at the literature on the amygdala, you see that it's not quite clear exactly which computations or behaviors the amygdala supports. It has been involved in many, many different things. And that's partly a function of the fact that we cannot really do causal studies in humans. There's a few lesion studies, but they obviously come with their own challenges in humans. And then there's a lot of animal work, but then especially in the affective domain, it's sometimes hard to translate that to humans. So we were wondering, is the amygdala involved in these processes that I just described, the emotional biases in approach and avoid behavior, where the amygdala has been involved using neuroimaging data previously, and also learning from feedback. So we went ahead and designed a task that can measure both of these things, and then applied TOS bilaterally to the amygdala to see if anything changes. The task that we used looks like this. Participants see a face on the screen with a certain emotional expression, and they have to decide whether they want to approach or avoid that face. If they approach, the face zooms in. If they avoid, it zooms out, and then they get feedback. So, opposed to many other studies who have looked at this, the right action is not instructed, but they just have to learn from feedback what the right thing to do is. And the task is structured such that for 4 to 7 phases in a row, trials, they see different faces, but the same emotional expression. So here's an example for 4 trials where they see happy faces. And then within this mini block, they kind of repeatedly try out whether they have to approach or avoid, and they get feedback, and they learn kind of over the course of this mini block what the right thing to do is. The feedback is probabilistic, so even if they do the right thing, in 20% of the time, they might see the other feedback. And then at the end of a mini block, the next face that comes on screen shows a different emotion, and we use 3 different ones: happy, neutral, and angry faces. And then they again have to work out what the right action is. And the right action doesn't transfer across many blocks. So if they've previously learned that they have to approach the happy faces, then the next time they see a happy face mini block, it could be an avoid mini block. Okay, so they have to try it out every time the emotion changes. They have to learn it. Participants are quite good at doing this. It's not a very difficult task, although there are confusing trials in there. But what I'm showing here is that they learn over the course of a mini block the correct emotion action mapping. So in the colors, you see the different emotions, and then in the dashed lines is when the right action is to avoid, and the solid lines is when the right action is to approach, and on the y-axis, I'm plotting the probability that they're approaching on average. And you can see that that they nicely separate after already 2 trials. What you can also see is that the colors are slightly different. So people do show an emotional bias in this task. They tend to want to approach the happy faces more and the angry faces less. You can also note maybe that on average, they're all, for all emotions, they're above 0.5. So they have a general approach bias for all of the faces. But then between the emotions, there is significant difference between them. Okay, so that's kind of the emotional bias that we can tinker with in this experiment. We also constructed a little cognitive model, very simple learning model that kind of perfectly recapitulates this average behavior. And it has a couple of parameters, has a positive learning rate, negative learning rate, a decision temperature, and then critically 3 different parameters that code for these biases to approach or avoid faces of a specific emotion. So this is just showing that you can capture the behavior in the task quite well. We also looked a bit at reaction times. The task was not optimized to do this, so that's why it's not our main outcome measure. But we do see some interesting things. So for example, at the beginning of a mini block, people tend to take longer with their decision. This is when they basically have to guess because they don't know yet what the right mapping is. But also over the course of the mini block, they tend to get slower. And we interpret this in a way that they, after a while, they know that mini blocks are 4 to 7 trials long. So once they're at trial 4 or 5, they might look out for a change in emotion to confirm, that they're still in the same mini block or maybe a new one. So the reaction time seems to scale with the probability that there's a new emotion coming up. And they are slightly faster for the happy faces, but that's kind of irrespective of whether they need to approach or avoid in this task. Okay, so now we want to change this behavior. And for that, we use focused ultrasound. This is a relatively novel stimulation technique in humans, at least. It has been used in monkeys already a lot and in rodents as well. And it is really just ultrasound waves that we're centering on a specific part of the brain using a transducer. The advantage of the ultrasound is that because it's ultrasound and it's not, magnetic stimulation or electrical stimulation, we can actually get to really, really deep brain regions and with a very high spatial precision. So all of these, deep, interesting, small nuclei in the brain that have previously not been available to reach for us for causal non-invasive stimulation are now kind of open for us to explore. And the field is quite young and it's evolving really fast. So there's a lot of things we still don't know about what this technique does, but there's also a few things that we already know. The ultrasound can have effects over a range of timescales. So we can use it in what we call online experiments where we stimulate, in a during specific trials, for example, or in a specific period of a trial, just briefly, and then switch it off again, and then look at the immediate effects. But we can also look at in what we call, we can also use it, in what we call offline studies. And this is where we stimulate for maybe an extended time, and then after the stimulation, we then measure the effects in, behavior, neuroimaging, etc. So it has also these more long-term plasticity effects. And if we kind of look at how it fits in with other stimulation techniques, you can see here that all of the green ones are the non-invasive stimulation techniques. And out of these, it is the one with the highest spatial resolution, or, spatial precision, and spans kind of a range of timescales. So it's really cool and exciting, and I hope I can get you on board with this. Okay, so in our study, what we did is we had participants, come for a phone screening, and then they came in for 4 in-person visits, where with the first visit, we just put them in a 3T scanner and acquired some structural images, and also a 0 echo time sequence, which is good for imaging bone. And that is important because the main obstacle with ultrasound is getting through the skull, especially through the human skull, which is extremely annoying. So we need to image it so that we can then plan individually based on a person's anatomy and also on their skull shape and everything, where we're going to hold the transducer to reach our target. And this takes 1 to 2 weeks. And then they come in for these 3 repeated visits, the order of which was counterbalanced, and we stimulated either bilateral BLA, or as a control region, the insula, or we just played a sound that kind of sounds very similar to the actual sonication. This is an offline study, so we did simulate both hemispheres for 80 seconds, and then afterwards rush them into the scanner as fast as possible. And then we let them play the task in the 7 Tesla MRI, and also recorded some resting state, task fMRI, and MRI state. In terms of planning our sonications, what we need to do is we need to run acoustic simulations to find out, if we hold the transducer here and use a specific depth, etc., where are we most likely to end up? And these are the kinds of maps we can get from that. And here I'm showing both the planned field and the actual post hoc field. And what that means is, from our structural scan in the beginning, we run these simulations and then we plan the trajectory. But then during the sonication, we also use a neuronavigation system and record the actual position of the transducer while we're stimulating. And then we use that empirical position of where we actually held it to run the same simulations again, but post hoc, to see where we ended up stimulating. And here the two, on average, overlap really nicely, so you can actually only see the red one. And they're kind of quite on target for the BLA. Now that's the group average, and you can also see that the intensity is roughly what we try to achieve. So this is kind of the acoustic pressure or the acoustic intensity at the peak of this field. And we were aiming for something between 5 and 8 watts per square centimeter, and that's roughly where we ended up in most people. However, we didn't manage to always hit exactly where we wanted in individual people. So here's an example for a post hoc simulation that looks really good. This is a really, really successful, if we can believe the simulations, stimulation of bilateral amygdala, exactly how we would like it to see. But then this is another example of someone we stimulated. And here you can now finally see this brown BLA anatomical mask that previously has been covered by the acoustic field, because in this participant, we completely missed the amygdala. Okay, and there are many reasons for why this happened. We've learned a lot during this first study that we ran with this technique. And I'm sure we can do better now, but I think it's also a useful learning experience and good to be transparent about these things, because that obviously influences, how reliable our results might be. And in fact, out of 30 people who completed all 3 visits, we completely missed at least one hemisphere in 9 participants. Yeah, so that was obviously a bit disappointing for us to see, but at the same time, we were glad that we did run these post-hoc simulations because now we have the information. And then we decided to only take the 21 forward where we actually did bilaterally hit the amygdala. And the reason that we think we need to hit the amygdala bilaterally is just knowing from lesion studies that obviously if you just take out one hemisphere, then the other one might compensate. So if you want to see a behavioral effect, we figured we kind of need to have hit it at least a little bit bilaterally. So we're now going to look at the remaining 21 participants. And before I look at the behavioral effects, I want to just briefly look at some neural data to see whether we did anything to the brain with our stimulation. And for that, we ran 2 resting state fMRI scans, one at the beginning of the scan session and one at the end. We also ran task fMRI. This is being separately analyzed and will be published separately. But we also ran MRS sequences, so spectroscopy, to look at EI balance and maybe get an idea of whether our effect is more excitatory, more inhibitory. I'm not presenting these data today. If you're interested, I can chat a little bit about what we find. It looks quite interesting, but we're not as confident in these results as we are with the resting state. So I'll focus on these for now. What we see there is that if we look at the connectivity of the BLA with its typical network, so those regions that it is monosynaptically connected with, then after amygdala sonication, we see a significant reduction in that connectivity, as if we're kind of taking that region out of its network. This was not very different between run 1 and run 2. If anything, it was stronger in run 2. So that was surprising to us, but it seems to say that we can see this effect lasting for more than 1 hour. And this is how the network looks like that we investigated. The regions are here sorted by the strength of their average connectivity across the sessions. So it starts kind of at 12 o'clock and then goes clockwise. The hippocampus is very strongly connected with the amygdala, and then it goes downwards. And on the right, I'm plotting the difference between the SAM conditions and the amygdala stimulation conditions. And you can see that the black line at the outside of the circle is kind of no change. And then for all of the regions that we looked at, there was a reduction in connectivity with the BLA. So we were quite happy to see that. So it indicates that we did do something to this region. Okay, so did we also do something to behavior? This is just a reminder. This was the kind of the main way of looking at the emotional bias in this task, is this probability to approach a face based on what emotion the face is showing. And after we simulate the BLA, we find that this is changed. People become more similar in how they approach happy and neutral faces. So the difference between the two is reduced. But this is mainly driven by a change in the approach probability of the neutral faces. Yeah, so the difference is smaller, but if you look at them individually, it's the change in the neutral faces that is driving this. And this is significant when looking at it as an interaction with a covariate that we designed for these 21 people. So based on our post-hoc simulations, we looked at in whom were we particularly successful, in whom did we cover a large area bilaterally of the BLA. And that result is also really, really nice. So here you can see on the y-axis the probability to approach a face. The dots are individual people, and then on the x-axis we look at the volume of the BLA that we stimulated bilaterally, and we can see for the neutral faces that this effect is driven by those people where we actually managed to get some acoustic energy into the BLA. For the happy faces, it kind of has a trend in the right direction, but it's not significant on its own. It was a little bit unexpected, this result. So we were hoping to see a change in the emotional bias, which would be defined, by the difference between angry and happy, or the difference between angry and neutral and happy and neutral. We didn't really expect this to be driven just by the neutral faces. And so we kept on thinking a little bit about what this could mean and why the neutral faces are special. And the thing that we came up with is that maybe neutral faces are just emotionally quite ambiguous. And so the first thing that we did is we looked at how people rate these neutral faces. So we have obviously a finite set of of face stimuli that we used. And it turns out that in the 20 or so neutral faces that we used, people experience them as significantly more happy than angry. Yeah, so we looked at the ratings that people give these images on average, and we seem to have by chance selected neutral faces that look to people more happy than angry. And the other thing that is also a little bit consistent with this is that if we look at the reaction times that people show when deciding to approach the faces of the different emotions, then we can see that after amygdala stimulation, people get slower, both for happy and for neutral faces. There's no change for the angry faces. So it is a bit speculative, but our interpretation is that it gets harder to distinguish these faces from each other after amygdala stimulation, and that kind of suggests that the amygdala has more of a role in emotion identification or processing in this task, rather than this behavioral steering of the bias. But I'd be very happy to hear your thoughts on this, because we're still figuring this out. Our effects are relatively specific. So the sonication to the BLA did not affect learning from feedback. And that is shown in this plot. So there's no difference between the sham and amygdala sessions. And of course, we also looked at our control region, the sessions where we stimulated the insula bilaterally. If you're interested in the insula, I can chat more about this. It was a little bit of a more complicated target than we had hoped. But when we look at this fingerprint again, this connectivity fingerprint in the BLA network, then we can see that the effect that the insular stimulation has is different from the one that the BLA stimulation has. So the red line is the BLA, and compared to sham, and there we see this reduction in connectivity. The blue line is the insular sessions compared to sham. And there was no significant difference. If anything, in some of them, it might be a little bit increased, but overall, certainly not the same as with the amygdala stimulation. The insular stimulation also did not have an effect on the emotional biases or the approach tendencies, but it had a different effect on the probability with which participants tended to repeat their previous action. That was increased after insular stimulation. And if we look at this across trials in a mini block and split it by feedback, so here the dashed lines is when they got negative feedback, and the solid lines is when they got positive feedback. And we see that it seems to maybe be driven more by the positive feedback. And this is confirmed by our model estimates of the learning rates, where the fitted model would suggest that the insular effect was specific to the positive learning rates. Again, nothing we particularly expected to happen, but at least it was nice to see that the insula, which actually is in a similar network as the amygdala, so maybe wasn't the perfect choice of control, but it has an effect on behavior that is different from what the amygdala does. Okay, so conclusions for this first study. The main effects we saw is that the TOS to the BLA did reduce connectivity of the BLA with its typical network. It did increase people's tendencies to approach neutral or ambiguous faces. One potential reason for this could be that after knocking the amygdala out or changing excitability in the amygdala, people had more trouble distinguishing between happy and neutral faces. And that effect scaled with how successful we were in stimulating. And then the ultrasound effects when stimulating the insula were different from the amygdala effects, in particular concerned people's tendencies to repeat their actions. All right, now I'll switch gears and think about the other problem. Oh no, sorry, I will not switch gears, and I will not yet think about the other problem. Instead, I will tell you that running an ultrasound study is quite mad at the moment, because a lot of these procedures are not yet established. It's getting better and better. But I certainly learned so many things during the study that I didn't expect. So we were running, we were testing our transducers in a big water tank with a hydrophone to calibrate our instrument, and spending days in this physics lab. We also, learned to run these acoustic simulations. The whole air prep when people come for the stimulation is a bit of a hairdresser experience. And our PI actually used this little 3D printed cooker on her hob at home to cook the gel pads that we then use for the transducer. So lots of things involved, and a really great team with Miriam and another postdoc, Johannes, Miruna as a PhD, and Tim as an RA, and lots and lots of other people who helped. All right, now I will switch gears and talk about this other challenge where emotions are dynamic and change, and we need to kind of deal with this. And a good example for thinking about this is, as I said, if you look at a face and you're kind of trying to figure out in what mood they are, maybe to decide whether you can currently approach them safely or not. You might deal with very different situations. So here, this young man seems to have, over time, different emotions, but it's not wild. Yeah, but you might also interact. And then if we want to estimate the mood and how the mood changes in this person, then we kind of want to look at the facial expressions that we've just seen kind of in the recent past to make our decision of whether we can currently approach them or not. And if the world is stable like this, or their emotions are stable like this, then we might want to integrate kind of over longer timescales, and look at how they behaved in the last couple of minutes rather than seconds. But if we're interacting with a guy like this, who has, strong fluctuations and only stays within one emotion for a short period of time, then this is certainly more volatile. And in this case, the recent past gets less informative about their current mood, and we might want to focus only on the most recent evidence. And the thing that I've drawn here on the left is what we call an integration kernel. So that just quantifies for a current belief or for a current decision, how much impact does the evidence have that I've seen previously as a function of time. And these kernels, normatively should get steeper if the world is more volatile, and they should get more shallow if the world is noisier, because then we kind of need to integrate over longer to get a more precise estimate. Okay, so this is what I wanted to study. Now, volatility and noise have been studied in the context of inference, but they haven't actually been studied that much in the emotional domain. So typical tasks that someone in the audience might be very familiar with are these, predictive inference tasks, where you have the means of predicting where something is going to happen, and then you see the outcome, and then you can decide how much you're going to update your prediction. And there's also a version of this game around in this circular arrangement where you're moving this little yellow paddle here along to indicate where you think the next coin will drop from the center. And then you see the next coin, and then you can adjust your shield. And in these tasks, it's really nice because we can scale, how often the mean changes, and we can scale how noisy the samples are, etc. But what I really didn't like about these tasks is that they come in this very artificial trial structure that most of our lab tasks come in, right? And I don't like that because, for one, decisions in the real world are not like this. So in the real world, we're obviously, confronted with this continuous stream of sensory input, and no one tells us, now the trial starts and now you have to pay attention. But instead, we have to look out for changes in these dynamically changing environments, and not miss the right time to act. But also, trial-based tasks tend to be a really inefficient design when we're interested in the neural signatures of all of our cognitive processes that we're looking at. And the reason why it seems really inefficient is that in this trial-based task, there is actually only a small fraction of time in the whole experiment where the participant is actually engaged in the thing that we're interested in. So, for example, the decision formation process. And if we record, for example, EEG data, then the rest of the data we can basically throw away. It doesn't tell us anything about how people make decisions, because it's just, breaks or feedback, other processes. So then we end up having to record for hours and hours because neural signals are noisy, obviously, and we need, lots of trials to get a good response. So that seems a bit unsatisfactory. And so in this area of perceptual inference, we have recently introduced a new task, which is very much like the circular inference task that you've just seen here, only that it is much faster. Okay, so this is the game that people play in our lab, and we call it the Save the World task. And I'm just going to play a video of someone doing the task, and I think it's very easy to understand what participants' task is and what's going on. When I'm doing an in-person talk, I usually ask people now what they think is going on, so I think we can skip that in the online setting. But it's basically the same as with the circular inference. So the participant is moving this shield along the circle and can decide to either move clockwise or counterclockwise. And their task is to catch as many of these beams that are shooting from this evil radiation source in the center, and that are going to harm Earth if they don't protect it with their shield. So whenever they don't catch a beam, they lose a bit of money, and that's what's displayed on the right. As you can see, it's super fast. The beams are coming quite fast at you, and you can continuously make your decisions and move. So within a short time, we get lots and lots of responses and lots and lots of pieces of evidence. And that's what we think is really good about this task. The other thing is it doesn't actually need any introduction. People can play it right away. They don't need to practice, and it's quite intuitive. Just to briefly show you that we can measure the things that we're interested in before I move to the emotional task. So we can see, for example, that participants in this task are faster to adjust their position to a new mean when they're in this more volatile context. So when there's more mean changes, and they're slower to adjust to a change in the mean when the noise is higher. So when the beams spread more widely around the current mean. And another way of looking at this is estimating these integration kernels that I showed you previously as a little graphic. So here we again quantify how much weight they place on the evidence that they see just before they make a button press. And we can see that there's this recency effect. So they place most of the weight in their decision on the most recent sample, which makes sense in this fast context. But the slope of these kernels is significantly different between the stable and the volatile and the low and the high noise conditions. So noise makes the kernels more shallow, and volatility makes them steeper, which is exactly what we were looking to see. Okay, but the actual advantage that the task brings only shows up when we look at EEG. And I also just want to briefly showcase that so that you understand why we're doing the other task in the emotional domain. So the main advantage of this task is that now we have these beam events, and they are coming in super fast. So within a couple of minutes of playing this task, we have thousands of events, basically thousands of trials. You might say, oh, well, but the neural response to these super fast events will overlap in time. And that's true. And that's the reason why we traditionally use these slow trial designs. But there are very powerful statistical techniques that we can use to unmix them. It's an approach called deconvolutional general linear modeling. Some people call it regression ERP or temporal response functions. It has been invented many, many times in the EEG domain, and it works really well. And it just relies on the fact that the temporal distance between the events is jittered, okay, so that they're not coming in at fixed intervals. And that's what we implemented in this task. Yeah, so then we can unmix them, and then we can actually look at the EEG signals to each of these beam events. And I was really, really struck by this. So what I'm plotting here is the average response in the EEG to a beam event. It's not a very interesting regressor, yeah? It's just like a visual evoked potential or a P300 or something. It's just the average response to each beam event, but I'm plotting it for 12 example participants, and the two lines are when they come in and play this task for the first time, and then when they come in again 1 week later and play this task again. And for estimating these, I have only used 6 minutes of them playing the task. And I find it quite remarkable that they are very different across participants. And in some of them, it looks like if you only saw one of these lines, you might think it's only noise. But then if you measure the same thing again 1 week later, the two small peaks that you see completely overlap. So that was really cool to see. And then we obviously wanted to know, does this also work for more interesting regressors that do not only just capture, there was a new beam event, a new visual event, but for example, how much evidence does the current beam convey? So how far off is it from my current estimate of the mean? Or also just, how different is it from the previous beam, which we could call a sensory change. And then we use other regressors, but I'm just showing you these two. We can measure the EEG response that scales with this evidence and with the sensory change, and find nice potentials for this. We also find that these amplitudes are significantly modulated by the volatility and the noise in the environment. But most strikingly, if we extract the mean amplitude of these peaks, then we again basically only need 6 minutes of data and can already measure these with super high test-retest reliability. So after 6 minutes, we're already above 0.75 in the intra-class correlation coefficient. And then the more data we use, the better it gets. So that was really nice to know. And then obviously now we want to do this in the emotional domain as well. Okay, so then we thought about how can we, measure how people integrate different emotional stimuli over time in a volatile environment. And the task that we came up with, it may seem slightly weird, but it worked. So the cover story was that there's 2 new movies that have recently come out, and people are interested in understanding the emotions that these movies elicit in the audience over time. Okay, so from scene to scene, what is the average response in the audience? And so we asked participants to look at a sequence of emotional expressions of different people and rate the average emotion in those phases, and that could change over time. Okay, and it was different phases because we're not interested in how a single person reacts to the movie, but kind of a group of people. And we asked them to rate either the valence or the arousal in these phases, and this is what it looks like. So you can see that it's a bit weird in the beginning, but you kind of see this really fast sequence of emotional expressions. And then you've got this slider bar at the bottom where you can indicate your current rating. This is an example for a valence block. And what you're seeing here is kind of the true underlying mean in this rating, but obviously participants were not, were not shown this, but they had to, adjust the rating themselves dynamically. We used, as I said, valence blocks and arousal blocks. So they rated valence from very negative to very positive and arousal from low to high. And we showed the same movie twice, once in the valence and once in the arousal domain, and then multiple blocks of these. And that is because both of them in our sequence varied continuously but independent from each other. And this allows us to look at the effect that changes in arousal have on ratings of valence as well. The other thing that we manipulated is the volatility and the noise. We could not afford in this task to have another 4 different conditions. So here we kind of matched them up. So we contrasted one condition where the mean valence or arousal, true valence or arousal in the images was stable, but exerted a high noise. And with one condition where the mean was changing more often, but the individual values were slightly more precise. And that is to kind of maximize our power for finding these differences in reaction times and other measures, because low noise should do the same as high volatility, as we've just seen in the other study. Okay, so we had this 2x2 design where we manipulated the statistics of the environment and then whether they rated valence or arousal. And it was really nice to see that we see similar effects both in an in-person sample and in a slightly larger online sample for how fast they are able to react to these mean changes in emotion. By the way, the task is much harder than the Save the World task with the beams. And a lot of people came out of the task and said they don't think they understood what was going on or rated anything sensibly, but they did. So if you look at how they rate, they roughly follow the true mean. So people are better at this than it feels. So they are faster in this more volatile and more precise condition. We can also look at something like these kernels. It works a little bit less well in this task for various reasons that we can discuss, and we're still kind of trying to find the most sensitive measure. But if we look at the online data, we do see a significant effect of, the stable condition showing a slightly more shallow kernel than a volatile condition. But this is a bit less beautiful than with the other task. The other thing that this task allows us to do is now look at whether people change their emotion processing or emotion rating behavior with valence or different directions of change, and we can frame this as an emotional bias. So we found, for example, that people are faster to detect changes where the valence switches towards a more negative valence, the average, and they were slightly slower to detect changes towards a lower arousal. Okay, so maybe to summarize, they're faster to see when the average valence gets worse, and they're faster to see when the average arousal gets higher, both in person and online. And the other kind of emotional bias measure we have is their rating errors. And it doesn't show in these plots extremely clearly, but what we find is that they're significantly worse at judging the positive category. So they underestimate the positive emotions more than they underestimate the negative emotions. Okay, so this gives us kind of a toolkit that this task can measure different things in the emotional domain. But now, what are we going to do with this task? How are we going to use it? This is my favorite part of the talk, because now I get to talk about my ketamine study. And I have been slightly obsessed with ketamine for the last couple of years, scientifically, obviously. And the reason is that ketamine has this really, really weird dual role in psychiatry. On the one hand, we use this drug as a pharmacological model of psychosis, because the acute effects of ketamine kind of mimic these psychotic experiences and the way that perceptions change. But on the other hand, we also use it as a fast-acting antidepressant, because the slightly delayed effects of ketamine are a significant reduction in mood in depressed people who are otherwise treatment resistant. So we've known this for a long time, but really we kind of have no idea whether these two effects are linked at all on some level of mechanism, which is quite surprising. We don't know whether we could design a better drug that is also an antidepressant, but does not people make psychotic during the infusion. And I think it is kind of reflective of this more general disconnect between the levels at which we describe the symptoms, at the cognitive and behavioral level, and the level at which we understand the drugs, which is at the level of synapses and circuits. So I kind of try and link these levels with my little models. And if we think about the cognitive mechanisms by which ketamine could exert these effects, then we look at the perceptual side of things. There's a lot of literature suggesting that ketamine might change perception by kind of changing the balance between prior expectations and new sensory information. This is what this equation in the middle is describing. And there's actually a lot of really consistent literature that this might be a good explanation for how it's changing perception acutely. If we look at the antidepressant side of things, it's much less clear. It's also much less clear that the main effect that's driving the effects on mood has to do with NMDA receptors at all. But there is a literature that indicates something maybe similar, where ketamine is different from the typical antidepressants that take a couple of weeks to exert their effects, and that are thought to work via changing learning and emotional biases in like perception and attention and so on. But ketamine works within a few hours. And so the idea is that it kind of weakens the impact of already existing negative schema or expectations or memories. And in a way, computationally, this is actually quite compatible with each other. So I have this little pet hypothesis that maybe ketamine is doing something in the perceptual domain and in this more affective, reward or emotional domain that is actually quite similar, and maybe there is a link between these different effects. So that's what I wanted to test, and that's where my tasks come in really handy. The way that the study worked is that we had, again, a screening visit, and then people came in twice for a first infusion visit, and then 1 week later for a second infusion visit. So they got ketamine and placebo infusion counterbalanced across people. But then each of the infusion days actually consisted of multiple days. So before the actual visit, they played our tasks online to train for 3 days. And this is when we kind of induced expectations in the various domains. On the infusion day itself, then participants came in for a really long visit of 7 hours or so, where we measured some EEG before they got the drug, during the drug, and then again 2 hours later. And this is where we obviously look at the effects of ketamine on behavior and EEG. And then a day later, they again played some of our tasks online to also look at the effect at 24 hours, because this is usually when the antidepressant effects peak. And then we had the same thing again for the second visit. We gave a relatively high dose of ketamine, which means that some people also got a little bit sick, which I'm very sorry for. But it meant that we could measure our task for a slightly longer time, so it's 60 minutes where we could let them play our task. And this was done in healthy participants, 30 people. The study is complete. However, to disappoint you right away, I'm still blind to the drug labels. We're about to unblind, but we're kind of currently still pre-registering our hypotheses for each of the tasks. And because we include multiple tasks, it's a bit of a coordination between different people. And so it has taken a little bit longer. So I'm really sad, but I won't be able to present the results. But I can just briefly, and then I'm also done with my talk, and just briefly walk you through the logic of what we're doing. So as I said, we're measuring the acute effects of ketamine during the infusion visit. And then during the infusion, they play this continuous inference task to capture how ketamine changes perception and the influence of these expectations about volatility and noise on how people integrate evidence. And then in the background, we also play a mismatch negativity task because that always works. And then 2 hours later, they play the faces task where they have to continuously emit these emotion ratings with exactly the same idea. So again, they have to integrate stuff over time and need to watch out for changes, but now it's in the emotional domain. And they also do an interoceptive task with EEG. And then 24 hours later, they play 2 decision-making games that are in the reward domain, where they trade off efforts and rewards and where they forage under threat. For the 3, so the 2 continuous tasks that are presented to you, and for the effort task, we use this online training to induce expectations so that then we can look at whether ketamine really weakens the effects of expectations on emotional evidence integration. All right, this was another study that was slightly mad for other reasons, but because of the infusion, we needed to run the study in a hospital setting. So we brought our little tray with all of the EEG and all of the cables into this room. And I'm really glad that we had a good active EEG system and that our signals actually look reasonable, because this room was full of electrical interference and lots of other challenges. And again, it was Maruna who helped me complete this task. Okay, so that's what I wanted to present. I hope it didn't take too long, and I'm looking forward to your questions, and thanks so much for your attention. And finally, what Søren already mentioned, I'm moving to Osnabrück. So if you're interested in any of this or the modeling aspect, do get in touch because I'll have a position there. Thank you.
Thank you.